Okay, so next up, are you wired?
I hope so.
Ready to go?
I hope so.
All right, good. So next up is Dennis Hansell from University of Miami, and he's going to talk to us about dissolved organic carbon in the Indian Ocean and give us, I think, a basin perspective, which I'm really excited to see.
Good. So thanks, everybody. Nice to be here. Thank you, Jerry, for the invitation. It's always good to be at the OCB meeting, seeing old friends, making new friends, knowing the community well, especially for young scientists here. This is really important for you to get to know everybody, become part of, the community for a long period of time. Maybe after 50 years, you can sit back like Frank Malero and say, seen it all. Just quietly back there. So, Mike's talk was great because, the dynamics of the Indian Ocean are really, he's shown us how much is being understood there. But that takes a lot of data, right? And as soon as you get out of the physical realm and get into the biogeochemical realm and the biological realm, then, there's a lot less data out there. So, it's an embarrassment of, paucity of data, really. So that tells us how important, just those observations are, right? The other thing that, the value of coming to this meeting is because you're made aware of what's going on, things you didn't know about, just to read a, poster title and that sort of thing. And the Noctiluca, mixotrophy that Joachim and Helga are talking about in the Arabian Sea for the last 10 years. I'm going to show you data from 20 years ago when Jagos was there. And, it was a really clean system and, it had lots of good export going on and, made some, you thought you could make sense of it. And now we're talking about predictability and exports, and all of a sudden this Noctiluca bloom happens, and Helga thinks it's anthropogenic, forced. How predictable would that have been? Are we anywhere close to it? And if we understand the system at one time, just a few years later, it can have a complete regime change, which is a little frightening. The biology is quite an issue. So anyway, Jerry asked me to talk about broadly carbon in the Indian Ocean, and I said I need to narrow that down a little bit because there'd be books and books on that, and so and they need a title fast, so I threw this title out there. I'll try to go beyond dissolved organic carbon. That's what I do with Craig a lot, and so we'll just do what we can. And then, this is out of the International Indian Ocean Expedition 2 science plan, I guess, and theme 6 is unique biogeochemical ecological features of the Indian Ocean. And that's what makes it interesting, is those unique features. Because a lot of the Indian Ocean in some ways is, you could be in the Atlantic or the Pacific. What makes it unique? And those are the kind of things that I'm drawn to. All right, so, just to help you understand what's out there, if you haven't downloaded GLODAP 2, you should. It's a wonderful, wonderful resource, lots of data. Mostly these data, hydrographic, organic carbon system, CFCs, nutrients, and that sort of thing. But if you want a broad understanding, snapshot kind of view, you get into these data sets. These lines are typically occupied, every 10 years or so, if they're part of the repeat hydrography program or GOSHIP now. So you're not going to get a big temporal sense, but they're a snapshot of what the system looked like at the time. Incredibly valuable resource, and when I teach undergrads, I have them download these things when we work with the data. It's really very powerful. As far as where we have, Craig and I generate DOM data, where do we have those data? Not many. I had very, very few data. This is where we have them. Jagos, that's up there, in the Arabian Sea. A couple of lines, we did, a bunch of occupations there in the mid-1990s. But that's a really nice system up there because of the monsoon dynamics that Mike described to us, hypoxic basin. It's really the monsoon dynamics that drew us in there, that Sharon Smith led the U.S. charge to get in there, and it was really motivated by alternating system biologically and biogeochemically, and it's really powerful in that way. These are all the lines, I-08 north, I-05, 6 south, and 8 south, and I think I'm missing one. And once you get the lines, then, again, there's not a temporal component to that unless 10 years apart is good for you. But, this line, I-09, these data that I'm going to show you here are from 8 or 9 years ago, but, Craig was just and the rest of the Ghost Ship team were just occupying that line last fall, I think. So there'll be new data coming out of there. But when you get up in the Bay of Bengal, you can start, hoping to get a sense for the importance of those rivers. Pete Raymond talked about the rivers, the globally important rivers, and those are some of the really important ones for, as he suggested, the inorganic carbon system, and another hypoxic basin. So really important for nitrogen dynamics, important nitrogen sinks up in those systems. Let's see here, there we go. The line that we have, you can see the Indonesian through flow there, so we'll just touch on that, take a look at what kind of a signal is there, and it's different than what I would have expected based on other observations. If you want to talk about a gullus leakage, a lot of carbon that's coming through here, it's part of the overturning circulation. So what part of the DOC that is being removed in the North Atlantic is coming here and being made up for through the gullus leakage, coming from the Indian Ocean. So you can start looking at these kind of bigger global scale questions with these sorts of data sets. And then the subtropical gyre, that's where we have overturning circulation going on in some parts of that, and DOC with that, DOC export taking place. So we'll just take a look at some of these things as we tour around the Indian Ocean today. And I'm going to start with the Jagoff's data set, and it apparently looks completely different now as far as the dynamics, according to Helga and Joachim. Again, these are the data sets, a lot of temperature and all that sort of thing. If you want biological variables, go to the Jagoff's data set. So for those of you that are in the younger side of this crowd, it's incredibly rich data out there. You just have to dig in. There's a whole series of deep sea research series 2 journal, dedicated volumes to the Arabian Sea that you can learn a lot about the system in a hurry, and then use the data to advance your own questions. Okay, so let's just start with some Jagoff's results that, I think it's a beautiful system. I feel so fortunate to have been able to work up there. There were two lines of interest, this north line and this south line. I'm mostly going to focus on the south line. It seems to be a little easier to interpret. So what's going on out there? Mike gave you some basics on this, and I'll just go over it real quick. Southwest monsoon is a really important time. You get relative low pressure system up here, relative high pressure system down in here, and the winds fly up there like crazy. Very strong winds. And of course, because of that, you got the waters being pushed or pulled off of the coastline, and you have coastal upwelling, right? And so you get these filaments coming off the coast of Oman, driving these big blooms, right? So it's a beautiful system, classic that you'd see off of the west coast of California, Peru, and that sort of thing. So very nice. During the southwest monsoon, that's a summer phenomenon. And then northeast monsoon, it's the opposite direction. So now you have the relative high up here, relative low up here. And so now you've got these cold, dry winds coming down here. And it has a different effect on the ocean. This is like the big cold fronts that come off of North America over the Sargasso Sea and drive convective overturn at the bat site, for example. So what's happening here is very much like what we would see every year at the bat site. What's happening here is very much like what we would see off of Peru, places like that. And in this one location, you switch one from the other, one from the other. It's an amazing opportunity to see that complete change in the system. And so here you have, again, it's just a convective overturn throughout the system. So you have a different, response as far as the biological response. Here it can be more intense because of that, really nutrient-rich water being brought up. Back in those days, it was diatoms and phaeocystis really dominating the production during the southwest monsoon. And according to Liu et al., Synechococcus made up about 80% of the primary producers or primary production during northeast monsoon conditions. So, these little tiny guys, little cyanobacteria, versus these guys, much bigger. So they're going to have, presumably different impacts on export production, the biological pump and all that. So again, nice system for contrasting those environments. So let's just take a look at the nutrients. And again, focus mostly on the south line. Northeast monsoon up here, the high nitrate waters are down here at about 80, 100 meters, and it becomes anoxic or suboxic certainly down there. And again, it's a convective overturn, so the water is turning over this way, it's reaching down, grabbing nutrients and bringing it up. And so you see this more uniform, low concentration being mixed like this. And again, this is what we see at a place like that. But here it's much of this line, because those winds are coming down here, cold, pulling out the heat, destabilizing the water column, and mixing it that way. If you're down here, 10 north, you'll see that it's just oligotrophic all the time. That was always our reference, oligotrophic water. It responded just much like the rest of the line, so it wasn't too effective as a reference point, but it did stay nutrient-free throughout the system. And then here's the southwest monsoon condition again, same line. It's just bringing up those nutrients really strongly and then bringing them offshore. All right, so very, very strong contrast in the mechanism of bringing the nutrients into the system. And these two really important systems as far as the biological response are separated by intermonsoons. And so the fall intermonsoon leads into the northeast monsoon. Northeast monsoon is a northern winter event. This is more of a, this is more of a fall, our fall, northern fall. And then spring, of course, spring intermonsoon. And so you've got these really oligotrophic waters, except for that little station there, throughout the system. And then, you bring nutrients up, come back to oligotrophic, bring the nutrients up, come back to oligotrophic. So it's really oscillating beautifully out there. So you can learn a lot about the biological pump in that system. So the question then is, this is what Jagoff's question, what's the response in the organic carbon, or the carbon field in general? And I'm going to focus on some of the organic carbon results here. Here's some data from John Mara, and it shows the primary production during the northeast monsoon, southwest monsoon here, and here are the intermonsoons, fall intermonsoon, spring intermonsoon. So again, more oligotrophic conditions during those times, and this is at a mooring, right? So he had optical sensors and some, I don't remember the details of this, but he was getting production off of that mooring there, and you can see that, according to these results, primary production during this time, the northeast monsoon, we just have that convective overturn, lower nutrient concentrations, Seneca-coccus dominated, is really very similar at that site to the southwest monsoon. Very different system, again, more diatoms, phaeocystis, and that sort of thing. So high levels of primary production at both of those times. And now I'm going to walk you through, again, on that south line, just the amount of carbon in the system. Okay, we're going to start in the spring intermonsoon. And all you have to do is look at the color. If it's orange, hot looking, red, it's got a lot. And if it is cooler, it's got less. And what we're going to do is walk through the seasonality of the organic carbon that's up there resident in that upper layer. And here's the end result. We're going to see this big oscillation from the spring intermonsoon through the, a year later into the northeast monsoon. We're just going to work our way through it, just a little cartoon kind of an image of what's going on there. So in the spring intermonsoon, we got, it's fairly hot, fairly high stock there. Oops. And then we have now, the spring intermonsoon is going to be followed by this southwest monsoon. It's kind of high here. You can see the upwell in here, so you're bringing up lower DOC waters and pushing it away there. So it's still pretty high. So here we are in the middle of the southwest monsoon. But then we come into the late southwest monsoon, and you can see that it's dropped out, right? So these are really pretty low concentrations relative to what it had been during this very oligotrophic period. Come into this very productive period, but no change in the system. Get late into the southwest monsoon, and things are dropping out of the system, and so we see the stock drop off. We come into the fall intermonsoon, and it's a remnant of what was there at the end of the southwest monsoon. There's not much of a change in the system, still oligotrophic here. And we come into the northeast monsoon right here. All right, so now we're in the northeast monsoon. We're starting to build that carbon back up, right? We're headed back to the intermonsoon that's going to be following this one. And then finally we have, late northeast monsoon. So we have this big oscillation. Spring intermonsoon is high because it was set up by the productivity in the northeast monsoon. And then it's all offset, removed during this period here. If we just back up, we see this oscillation then. Northeast monsoon, you build up the stocks. It's held during the spring intermonsoon, oligotrophic period. It's kept during the early southwest, and then sometime in that time, it just drops out. The bottom drops out from under that system. And then the northeast monsoon returns, and you build it back. So these are the data, not cartoon-like I just showed you. And it's for two different systems on that line, sort of the middle of the line and then down here, which is, again, our oligotrophic end member down there. But they both behaved exactly the same, which was, interesting and unexplainable still for me. TOC stock varied by about 2 moles of carbon per meter squared. That's a lot. POC, this is, again, TOC. POC was only about 0.3, so most of this variability was DOC building up, DOC being removed, DOC building up, DOC being removed. So a lot of variation in the system. And again, net primary production, according to Lou et al., was Synechococcus, really dominated by Synechococcus. So they perhaps had a major role in this recovery of building up that DOC. What we'd like to understand, what we don't understand, is that removal process there. So let's think about that removal process a little bit. So here's that same data set of the oscillation of the TOC stock, right? So here's the late southwest monsoon drop, fall intermonsoon, and then the recovery in the northeast monsoon. And this is mass flux, from Susanjo's trap data. And not a lot of flux really during these periods of the northeast monsoon. Maybe Synechococcus is not being exported to great extent. But then you have a massive flux, and again, it's mass flux, not just organic carbon, but all sinking material. Right in there, when we have the collapse of the organic carbon stock, remember a lot of it's dissolved, so it's not going to be part of those sinking particles, you wouldn't think. But when this big export event is taking place, then you also have that big loss of the DOC pool. And so what have we got? We've got northeast monsoon, convective overturn, low particle export, large net DOC production. So you have a response in the system with those modest nutrients, but it's in the euphotic zones kept up there. Southwest monsoon, you don't have that much of a response in the biomass, but they're going to be exported, right? So they don't accumulate as well there. And it's not the only thing being removed. These are data from Chris Measures and Sue Vink. And the blue just indicates the loss, this is negative values, the loss of iron between basically early southwest monsoon and late southwest monsoon. So they're out there measuring iron. It too is removed from the system. So you're losing iron, you're losing dissolved organic carbon, you're losing particulate organic carbon. What's the mechanism for all of that? So you can make things up. We have a lot of dust coming into the system, right? Southwest monsoon winds are picking up dust from around here, and it's a beautiful system. If you're on a ship out here looking back in here during the sunset, it's just red beauty, really, really nice. But a lot of lithogenic particles. Certainly they're going to be adding a lot of reactive trace elements. Those trace elements, along with upwelling, are going to allow the production of the diatoms and the phaeocystis. Now the DOM, I don't see evidence for these guys generating a lot of that, but there was a lot there from the previous season of the northeast monsoon, right? Synechococcus maybe put this into the system. And I'm just making this up because I don't know. If we start adding, I know Monica doesn't want me to say TEP probably, but some kind of a glue into the system, make these aggregates and bring it out. So I just don't know how else to do it to scavenge these trace elements. How do you scavenge DOM unless you make it part of that aggregate? Interesting system. We don't have the answer to that. Maybe some people who work to the system do have the answer to it, but I don't know what that answer is. So it's a really fascinating question. So these data, this is an incredible place, hence the reason JGOPS went there, for investigating processes, control, and export. We don't have all the answers, but it's a wonderful place for completely contrasting systems in one spot. Very oligotrophic, nutrient entrainment by convective overturn, nutrient entrainment by, additions by upwelling, and all the different extreme responses to all of those forcings. So very nice. And the fact that the mixotrophs are out there dominating the system now during the northeast monsoon, the carbon system must be completely different. So we need to understand that. If predictability is a goal, we need to get into these places that are so anthropogenically impacted that you have a complete regime shift in it. Okay, so we've got this, right? And so we've got this little cartoon of what's going on out there, and we've seen this during the intermonsoon. We're following the northeast, so we have high concentrations, right? You're following northeast when this stuff was produced. And then you come into the fall intermonsoon, we're following the southwest monsoon, so we have these low concentrations. And so the question is, do we see that kind of a response anywhere else in the Indian Ocean, beyond the Arabian Sea? How large scale a phenomenon is it? And so we can take a look at that, because some of those lines that we did over, those long lines over in the east happen to fall during these kinds of intermonsoons. And it's no coincidence we like to be on ships during intermonsoons rather than during monsoons. Okay, so what have we got? So we got two lines over here. So here's I-9 north, here's I-8 north over here. They're occupied probably more than 10 years apart, but as it turns out, let me back this up, sorry about that. Turns out we were out there during the northern spring intermonsoon of April 2007 and the northern fall intermonsoon of October 1995. All right, so these two lines, and they're close enough to each other to say, let's look at those. And then we'll put up this thing as a point of reference, right? This is what was going on in the Arabian Sea. And this is what we would, during the spring intermonsoon, we had this high stock, and we come up here to this data set. This is the spring intermonsoon. And here's the equator, just so we're up in the north here, northern hemisphere. And we see hot spots, right? We see those elevated concentrations. And we come down to this data set. Fall intermonsoon, it's here in Sri Lanka, so there's not a lot of northern hemisphere to look at, but clearly it's different than that. So it tells us that there's a zonal synchrony in the system. A large part of the system is responding to those monsoon forcings, right, in a common kind of a way. So, that's nice that it's not just the Arabian Sea. We learn about that, but we don't know anything about the rest of the system. It looks like, the forcing has some commonality across the system. The odd thing, though, is that we have zonal synchrony, but we also have meridional synchrony, right? So where we have, let me see if there we go. We have high values here in the northern hemisphere, okay, and we say, well, that's because we're following the northeast monsoon. Well, what about this? Now we're in the southern hemisphere. It looks just like the northern hemisphere, and then we come down here and say, okay, this is low because we're following the southwest monsoon. Well, what about this? This is kind of odd to me because, these are very far apart, northern hemisphere, southern hemisphere. They shouldn't behave that way. Now, I think it's just by coincidence. I think I can rationalize this, seasonality and say it's just spring, winter, summer, that kind of thing. You just explain it that way. But it is kind of interesting to see that and wonder if there's more than just coincidence involved, sort of far field impacts of those monsoons. Okay, so this is a model distribution of DOC in the system, and basically you could think of it as, if you're a more stratified system, then it's going to be relatively elevated, and if you're down, in the more polar waters, then the concentrations are much lower. Right? We've known that for quite a while that that's the case. And so if you want to export DOC, and that's what I want to pay attention to here, then you need to have DOC at the surface so that you can bring it down. And so if you're moving these waters down, then you can export DOC. If you're moving these waters down, then not so much, right? The impact isn't very significant at all. So you have to have these lower latitude waters being brought down. So we can take, this is salinity distribution at the surface. And just to give you a sense for where the major water masses of ventilation is taking place, there's some mode water over here, some subtropical mode water here, subantarctic mode water here, Antarctic intermediate water. So you can tell, if that DOC is low down here, lower DOC, that intermediate water is not going to be exporting much. The Persian Gulf water, Red Sea water, these are all, this high salinity water working its way into these systems, being made more saline. Some winter coolness, cooling takes place, you remove some heat, that water drops and then comes flowing out at depth. And so you can find, for example, Red Sea water down in this system, and it's going to have exported DOC because there's a lot of DOC in those surface waters, right? And so anything that's being formed water mass-wise at these lower latitudes is going to be exporting DOC. We don't have a good observation of them all, but I'm just, I know it's happening. If you're exporting at these higher latitudes, then you're not going to see that effect. So we can take a look at some of this, get evidence of it by looking at that line there, I8. And here it is. Here's salinity on that line. Here's the south. There's the equator. Here's salinity. And so here's that high salinity feature that we saw in the subtropical gyre, and we can see that high salinity water being shoved down there. So that's that subtropical mode water working its way into the system there. It's impoverished, right? Nutrient impoverished, so there's no preformed nutrients in that system. You can see that working its way. It's warm water, so CFC concentrations are moderate, but you can use those as a tracer for that water as well. And then there's DOC present at the surface, and so that's going to be exported. And so we can see that export going on in the system. Now, if you're higher latitude, as I was saying, you're not going to have as much DOC there. It's colder water, so you're going to be more enriched in those CFCs, so you can see that. But there's not a lot of DOC present, so you don't export DOC. You do export nitrate. So here's your preformed nitrate that is being shoved into the system because of the iron limitation of the system. Okay, so the interesting thing about DOC, it's something that Craig's been working on for a lot, and is the question of how does it accumulate up here? It's not available to microbes. It accumulates. You let the microbes go to town on it. They don't go to town on it. They can't even use it. But as soon as you mix it down, export it with that overturn, then it's consumed. All right. So here's our conservative tracer, showing us that high salinity water is being brought into the system. That high salinity water has DOC in it, but the DOC doesn't survive. So this is more of a general question. This is certainly not just happening in the Indian Ocean, but what conditions allow DOC to accumulate at the surface? We don't know what controls net DOC production. Why is the concentration what it is? Why is the pool as big as it is? And then we don't know, what is it really that allows it then to be consumed once you remove it to these other microbes, these other populations of microbes at depth. So as far as predictability, we need to understand those mechanisms before we can make a lot of headway on that. Okay. All right. So I want to, just jump on to some rivers. Pete showed this yesterday. Pete shows Raymond and Spencer 2014. I think that's the ebook, and then I referenced the hard copy because I'm a hard copy guy, and so different years, same reference. And this is, the figure that I think he said Rob put together, and it says DOC flux in the 30 largest rivers, right? And so, this is just to give me a sense, what do we know relatively a lot about as far as these rivers? The Amazon's a well-studied river, right? There's people in this room that study the Amazon and the outflow, the impact on the biogeochemistry, within the river system and outside in the coastal system. The Mississippi, a lot of information there. The Arctic rivers, a lot of attention there. There's a real strong community of scientists looking at those because of the warming going on, the mobilization of the carbon in the permafrost, the organic carbon, really important system. This system, though, that's really important too, right? Look at the cluster of those major rivers there, moving a lot of carbon. Pete told us a lot of inorganic carbon is coming out of the system, but we know very little about that system. And so that's a problem for us, I think. It's harder to get in here because of all the national jurisdictions. That line that we go in there is all international waters. We don't go too far from that. A lot of rivers coming in. Here's the main group, the Ganges, et cetera. And here's the result. This is a figure of salinity distribution, southwest monsoon, so that you're bringing this high salinity water into the system and pushing this fresh water back up to its source. And so you can see these rivers right up in here are really important for that distribution. Now we know that they're going to be bringing in a lot of carbon, and I just want to real quickly explain. Mike showed the precipitation patterns. When you're in the northeast monsoon, remember those winds are pushing down off the continent, so it's pushing that intertropical convergence zone away from the northern section. So the precipitation is down here, but then you have the southwest monsoon. It's bringing that precipitation right up into the Bay of Bengal there, right? So this is really important. It's not so much over here in the Arabian Sea. Arabian Sea isn't getting that precipitation except for along the west coast of India there. So this precipitation is really important. We don't know its relevance to ocean biogeochemistry. So what's, here's the flux out of the Ganges, it's sort of a southwest monsoon time because that's when the intertropical convergence zone is being pushed up there. When you have high flow, that's when your DOC concentrations pop up. So that's when the movement, the transport into the Bay of Bengal is really cranked too. Let me just pass this. What's interesting, or what is missing for me is, if we go up here where we have low salinity water, presumably that's the river influence. We don't have any special DOC. We should. If there's a lot of DOC in that fresh water, it hasn't survived while the salinity is still, showing that signal. It'd be nice to know what that is about. Why, that DOC is being mobilized somehow pretty quickly. I'm going to step through this more quickly because Jerry wants me to wrap it up. This is the intertropical convergence of the Indonesian through-flow water here, right? So now we're right in here in this line. This is salinity, high salinity, and then this low salinity feature. That's the low salinity Pacific water coming through. Here's the DOC field. When I went out there looking at these, expecting, what would I see out here? I expected to see high DOC associated with this low salinity feature. It isn't at all high DOC. Why did I expect that? Because if you go over here on the other side where we have a lot of low salinity water, here's some low salinity water here, there's a really strong DOC signal. So I thought, when we're over here, we're just going to see this feature coming through. We don't see that, right? So this is the low salinity feature that gives us that. But this is what we have over here, right? Really low. So, it makes me wonder about, again, I was anticipating because of all this high precipitation that we see with the monsoons, that it had a special impact that I expected to be like this, high DOC. It wasn't, it's like that. So it makes me wonder, what's the role of rivers, what's the role of precipitation on controls of surface ocean biogeochemistry? You look at this low salinity feature here, it's massive. It's a massive area. And we've got essentially, we've got that line there, we've got the line there. There are really very, very few data of biogeochemical relevance throughout this system. So it's a real opportunity to understand those, especially with climate change. How is precipitation going to change? Okay, so the questions that I posed, this is the last slide. In the upper ocean system, freshened by both major rivers, right, the rivers up here, and rainfall throughout this system here, can we distinguish their relative impacts and controls on biogeochemistry of the system? Right now, we can't do that, right? But they're massively, the importance of those systems, the freshwater system there, it's got to be really strong, yet we don't know. I'd say we really don't know anything about it, but maybe someone does, I don't. What are the mechanisms controlling initiation of particle export and associated scavenging? That's the interesting thing. How do you lose the DOC? How do you lose that iron using the southwest monsoon as your model system? What processes allow DOC accumulation at the surface, and then what allows its consumption at depth? And then the final question for me is, when I see that export of DOC, I want to know where it came from. We're seeing it in the subtropical gyre. Where was it formed? I know that I'm forming it way up in the Arabian Sea. Mike showed us other upwelling systems, need new nutrients to form it. But, if the DOC in the Arabian Sea, where does that end up? Does it make its way down for export, or is it burned on the way down? Microbial, what is it? We don't have a mass balance kind of a look, budgetary look at the system. We're producing it. We can see it being produced. We can see it being exported, but do they match up? What happens in between? So those are all important questions for me anyway. Okay, that's the last slide. Thank you.
Dennis, do one quick question and move on to Jim. One quick question in the back.